What if we could eliminate cystic fibrosis by changing just one gene? Technically, that is how cystic fibrosis works. People with cystic fibrosis have a mutation in the CFTR gene, and this causes the CFTR protein to malfunction, leading to the symptoms that cystic fibrosis patients feel. One way to edit the genes of living organisms is to use a complex called CRISPR-Cas9. The CRISPR-Cas9 complex can cut genomes at precise locations. Now, before we get into the details of how CRISPR-Cas9 can edit human genomes, let's go back to where this all started: bacteria. CRISPR was originally found in E. coli bacteria. Let's say that this is the bacteria's cell membrane. In bacteria, CRISPR acts as the bacteria's immune system. Bye-bye viruses. CRISPR stands for clustered, regularly, interspaced, palindromic, short, repeats. Bacteria contain this DNA that is made up of short palindromic repeats, shown here in the green. These clustered sequences are regularly interspaced. Each spacer DNA is unique and it has a DNA sequence that can identify virus DNA. The spacer DNA is shown in orange, pink, and light green. Cas proteins contain helicases that can unwind DNA and nucleases that can cut DNA. When a known virus infects the bacteria, the CRISPR-Cas system can eliminate the threat by finding the matching sequence, then cutting the infectious DNA. Now you can take what you have learned about CRISPR in bacteria and apply it to any living cell.
One of the most popular Cas proteins is Cas9.
Scientists have modified the CRISPR-Cas9 complex to contain only two parts: the Cas9 protein and a guide RNA that contains the sequence that we are trying to find.
We know CRISPR can delete genes simply by cutting the sequence that the guide RNA matches to.
Cell processes will try to mend the broken gene, but this process is usually error-prone and the gene will end up deleted. Let's go back to the cystic fibrosis application. We need to cut the gene and then replace it. This requires a third part, the host DNA we want to insert into the sequence. In this case, that is a healthy version of the CFTR gene. CRISPR is still being researched. By editing one disease-causing gene, we could increase the chance of another disease or have many unknown consequences. But maybe one day we will live in a world where cancer is as easy to cure as a common cold.